just a bit there.
Yeah.
Clean it up. Thanks.
It's
That's
just
good.
it forms little golf balls.
Oh. And is that fairly common to see?
It is very common because it forms out of the water, precipitates out of the water, hydrogenic crust.
Is it common on many plates or just this particular area and plate?
I believe it's common on many plates.
If you hold that zoom, I'll try and get a little
Yeah.
closer here.
Yeah.
See if I can do the bug on the windshield.
There we go.
All right.
Oh,
What kind of
Nice
jelly
jellyfish.
do you think we're seeing right here?
wow.
I don't know the name of it specifically, but I think it's a type of helmet jellyfish, which is one of the true jellyfishes, I think. They're not like a hydroid medusae.
It's a stark grain size change.
Yep. That thing of Clorox wipes to your left there.
It looks like there's a little depression here.
Should be over to your left there. Yeah, somebody was eating ice cream at the console. Either that or Kylie's.
Oh, wow.
I'm gonna call these nodules, not nodules.
Yes, please. Unless you want to do a scoop, nodule scoop. I don't know if they've burned the scoop yet.
Maybe
No.
we'll...
What's that?
Huh? Was that somebody
Um,
downstairs?
I don't know if we have room to put it in the box with the...
Yeah, I mean, we scooped in the previous few dives, and they weren't nodules; they were just bits of basalt. And I don't think someone can correct me downstairs if they're listening. I think we could pass on this scoop option,
Roger.
unless.
no, I would pass,
They look like nodules.
but I'm not 100%. Kevin or Rob would know
They
better.
do look very similar to the. Got zinophyophores in here, maybe a few small bamboo corals, Bathypathes. So there's a fair bit of life in this.
Would you say this patch of ocean floor gets a lot of detritus, or is part of the water column?
A lot, I'm not sure, but it definitely gets, it definitely accumulates, probably very slowly.
So what else is common to see at this kind of the bottom of the ocean floor aside from sponges and corals? Is there any specific other like fish species or different type of crustaceans that we would normally see at this depth on these floors?
Squat lobsters we saw quite a bit. Sometimes you'll see isopods. I've never seen a giant isopod out here like we see in the Atlantic, but yeah, I suppose these depths would be right for giant isopods as well. But they typically need more sediment habitat.
Nice Farreidae sponge.
Yeah, Farreidae sponge. If there's nothing really high relief in sonar, we can move briskly across this
Yeah, we're
area of lower slope.
ship's moving all the time here.
Okay.
Do you want to increase speed?
What do we-
0.3 knots?
Yeah, we can do 0.3.
Yeah, we can do 0.3.
Bridge nav?
Speaking of squat
Can we
lobsters.
increase speed
is
to
a-
0.3 knots,
I
please?
believe this is Tritopleura, type of glass sponge.
Is that another Farreidae or?
No,
Okay.
well, no, I'm not sure. No, it's a Farreidae, yeah. There's a Tritopleura that looks very similar here, but it has more like long blades, as opposed to these frills. I would probably lean towards Farreidae for this one.
And how do you spell that?
Farreidae?
Mm-hmm.
F-A-R-R-E-I-D-E. double R-E-I-D. A-E is the family, Farreidae.
It's a good question, Matt. It's often hard to visualize some of these words.
Yeah.
How do you visualize a word?
You don't visualize words?
Would have been a good one for photogrammetry.
Yes.
It's not phonetic, definitely, which just means it's my worst nightmare.
The still cam's got Atalanta
Oh yeah.
coming down. I don't want to get much lower than that.
No.
I lost you.
Yeah.
Maintain double digits.
Yep. Maintain in double digits.
Took our turn out of our tether.
Very nice. Aesthetic and functional.
Oh,
That's what we strive to be.
Is that the mantra?
Oh, another sea cucumber.
Oh, good eye, right in the bottom of the screen there.
Oh.
We collected any of those this dive?
Yes, at least one.
Yeah, I think
Yeah.
we've got a couple actually.
I think we have 2, yeah.
Nice.
got time for a quick zoom there? I can hold it still.
Is that a
Still.
Bathypathes by it?
Still.
Or
Um,
Stauropathes?
Yeah, I would.
There you go.
Not
full
sure.
zoom there.
It's at a very
Yeah,
strange
sure.
angle.
There we go.
It's just perpetually out of focus.
Yeah, it's out of frame too.
So transparent.
Yeah, it does. It looks almost blurry.
You're telling me.
It's actually censored, so you can't see the insides.
Yeah, sorry, guys.
It's done on purpose. It's a family show.
Okay.
PG rated.
Nautilus is PG.
Bridge nav.
Can't be showing such
Can
scandalous
we move
things.
100 meters,
Such as
250,
sea
please?
cucumbers. Gross.
Thank you.
100, you're crazy.
I know. Going nuts.
Build up a good head of steam here.
Delta Dan and the benthic storm's going nuts.
Give it 30 seconds. We're going to stop to take a rock.
That's
Yeah.
the whole, that's the watch name.
Perhaps.
Delta Dan and the benthic storms. That's great.
And the what?
And the benthic storms. Oh. Who plays what instrument?
I'm so glad that's stuck.
So do we have any musically inclined folks on this watch? And for those viewers tuning in,
We know we have a piano player. Ah, yes.
I like to dabble. I
play
think
the-
Dan, I've heard Dan on the guitar. Video
Oh, yeah.
Dan, he's pretty
play
great.
I play the triangle too.
Ooh.
No, I'm kidding.
I can play percussion instruments as well.
Percussion?
Yeah.
I mean,
Who said that?
Ooh.
Brandon?
Brandon,
awesome.
what do you play? Percussion?
Well, I took 4 years of percussion in high school.
Oh, awesome.
Wow.
So I can play drums and
I play drums too.
xylophone and
Oh, that's cool.
timpani and triangle and tambourine.
What's the huge drums you hear at the back?
Timpani.
That's timpani?
Yep.
Okay. Those look, oh, I always wanted to try
The
those.
only bass cleft percussion
About as
instrument?
low as I want to get here.
Or one of the only ones?
Yeah. That's nice, actually. We're stretched out pretty far.
Yeah.
Those look really fun. Yeah, I'm so glad
This might
someone
be called a
brought a
ficus.
guitar.
Oh, hey, look, a question for me. Hey, Matt, how do you get to be a guest student on the Nautilus? You email them until you become too much of a nuisance to ignore, and then eventually they'll give you a position.
But seriously.
that the one we pirouetted around? It must be.
In other words, you show some perseverance.
Oh yes, exactly. Well, really, it was
Enthusiasm.
I started networking with people. I started sending out emails, and eventually I got to talking with some people, and then I was given just throughout. I just went up the chain of people like, oh, you should, if you know this person, here's someone that I'm going to introduce you to, and then I got to.
Yeah, I think
and
that's
over
it.
time,
Paragorgia, yep.
it manifested into this. So never
Looks
give
like it
up.
had zoanthids on it too.
We had a great conversation on that. I think it was two watches ago about how you got to your current position and what advice you give. And Kakui gave some really good advice that worked for me as well. And that's what it was perseverance.
Full send.
Full send.
Full
That's what
send.
it was.
Oh,
Thank you.
rock.
Thank you.
Rock.
Oh, rock. Oh, rock. Okay. Nice holler.
See a rock you really like?
Yeah, if this is like disconnected right there, which might not be. It's
No. I should
No.
be able to catch him on the fly. Wow, I gotta turn this.
Can you give me a zoom right there?
Yeah, of course.
Yeah, we definitely want to zoom first. Sorry.
Oh, sorry, Dan. You can focus on her
Oh,
for
yeah,
now.
no worries.
Go ahead. Zoom in there.
I can zoom both of you guys.
I got it now. We're good.
Yeah.
Might be separated.
yeah, I have no
Give me
idea.
5 on the.
It's a good size rock.
Yeah, and it has a good
5 on
uniform
the
shape.
bubblegum, please.
Hopefully it'll fit.
Coming over to 5.
Is this one you want to use for like dating the
Do
seamount?
a couple of in situ
Yeah.
shots.
Okay.
Like screen grabs. Just in
That's all
case
the
we do
rocks we're
sample
looking
this
for,
enough.
are
Okay.
dating seamounts.
So you're going to get it on board, and then what are you going to do next? How do you date it?
We have to cut it, check it out, take a thin section, look at the minerals in the thin section, characterize it. Oh, nice.
Wow.
Oh,
Smooth.
Like a glove.
look at that.
Yeah, I like that guy.
I
Happy
like
with
the
him?
brittle stuff.
Oh.
Got some
Chilling
bonus
down
watch
there.
bugs for you.
Okay, you can go.
Where was
on.
I?
Toy in the cereal box right there.
Yeah, we
think
characterize
it's,
it.
Starboard box.
So what does it mean to characterize a
Right.
sliced open?
So we describe
Basalt.
it.
Okay.
Yeah,
Go
so there's
on.
various ways
Full
to
zoom
describe
there, Dan.
a rock,
Full zoom, yeah.
Yeah, see what.
like mineral quantity in terms of percentage. If it's above a certain percentage, we can call it something rich, so olivine rich or something like that.
Okay.
And there's also various
Starboard box there.
texture descriptive words.
What is this? This?
A nice view
CPX,
of Atalanta of the
A
bio box right
clinopyroxene,
now.
it's a mineral.
Oh, yeah.
A mineral we're looking for to date.
What?
Look at that.
Wow.
Where do you want this thing?
Set feed
what's-
two.
We can,
What's open?
we can try starboard box, CRD.
Sorry, I can't hear you.
Oh, it's good.
Starboard box, either CRD maybe.
I don't think it's gonna go in CRD.
Alright, we can put it in Omega forward box.
We could just drop it in the starboard box. It'll fit in
Your
F.
F?
Yeah. Can you put it in F?
Yeah.
There you go. Cool. You can fit a lot of rocks in there. Dan knows that.
Yeah.
Nice.
Stack them up till you can't get them in there anymore.
So just this one, I kind of would write, where it fell approximately. This one is like in the forward, like in this area. Yeah. So it's easier to tell them apart.
Okay.
And that's why writing information like any attached biology
really
is
nice
really
shot of
helpful,
from
because that can sometimes be a
Sweet.
really easy
Zoomed
way
in
to
on the
just
arm.
pluck the one you need.
Okay, then
So
you can go wide
the one
there.
with the brittle star. Even though the brittle star is not necessarily going to stay on there, sometimes they do. But we can always go back and look at photos on the seafloor and compare the rocks on deck if we ever have questions.
Nice, okay, sweet. Thank you.
Here's another question for our ROV pilots.
Can I finish
Oh, never mind. Go for
describing
it.
what I was doing?
Yeah.
So we characterize it with a bunch of different words that you guys wouldn't understand. And then,
You might have some geologists out there listening.
yeah, I know, but for you guys. They know what I'm talking about, those geologists. So we'll take it back to the lab. We'll crush it to the size dimensions we want.
Crush it or smash it?
Crush it. We have machines that crush it.
Really?
Yep.
What's that machine look like?
We have a couple of them. There's one called a badger.
Zoom there for us.
Got a wall right behind me.
One called a chipmunk.
Sorry.
And then a disc mill.
Hmm.
And the chipmunk looks like a chipmunk. It's got two big circular sides on it, like it has nuts in its mouth. Anyway, we crush it and then sieve it, clean it, acid leach it.
Sorry.
This is probably heteropathes, or yeah, I think heteropathes.
Acid leach it, pick the minerals out we want, or ground mass that we want.
Come up.
Send that to a nuclear reactor
Sorry, I thought
be
it was
irradiated.
on the other side, and I was waiting.
and then it comes back and we send it through a mass spectrometer.
And that's how you characterize the minerals that are in it?
No, that's how we date them.
Oh, wow.
Yeah.
Is it okay to handle them after they've been irradiated? What's the
Yes,
risk?
it's low radiation. I mean, you have to be trained. I don't do it. We have a lab manager that does it. But yeah, it's pretty harmless.
It's cool.
I don't remember the exact
I think
concentrations, but way below recommended.
very cool.
Yeah. And then with argon dating that we do, you have to do a crap ton of corrections, which I'm still learning, so don't ask me.
What kind of corrections?
You have to correct for various elemental stuff because argon is decayed into by potassium, so you have to correct for potassium, and you have to correct for calcium, and other stuff too that I can't remember, but
Gotcha.
Kevin would definitely tell you all about that.
Really bizarre USBL fixes here. I'm just kind of scooching backwards.
Yeah. Can we slow to 0.2 just on the hill here until I catch up?
Bridge nav. Can we slow to 0.2 knots, please?
Stauropathes
Thank
are
you.
the mustard colored ones.
Which ones?
Stauropathes, the mustard colored
What you looking at there?
corals.
Looking at the bios.
Quick
Is that
credit.
it?
I'm just
Yeah.
making notes for data logging purposes.
Oh, right. Yeah, let me-
Not requesting a zoom. There's a dandelion siphonophore right in the back of that crevice.
Oh,
That's zoom worthy.
And
wow,
another
yeah.
Okay, hold on.
prinoid looking
That's
coral
good
with
there.
another Stauropathes?
That's good.
Yes.
I can do a half zoom there. Till I
Oh,
get-
orange sticopathes in the
These guys
background.
have a really cool web.
Whoa, wow.
Sticopathes is STICHO.
Okay, I can zoom a bit more there.
Like Sticho. Stichopathes.
It's like a spider web.
Yeah. They're hard. They're
Yep, very well anchored.
just the
Wow.
thruster wash of the ROV.
And so
will
that's how they anchor themselves to the
blow them
substrate?
away.
Is by
Yep, yeah, and sometimes they have lures that they'll extend and use those lures to capture
Never
prey.
really gotten a
It's
good
like
shot.
Spider-Man. Weird.
Jeez. This one's
Okay.
clearly been there for a
get a
while
little
and has some
tug here,
pretty
sorry,
good
Dan.
stability structure. Hardly see it moving at all.
Yeah.
We have a question for the ROV pilots. How much training does it take to operate an ROV? What does one go through?
Well, I think that answer probably differs whether you're flying Atalanta or Hercules. I can tell you, I sort of learned as I was flying Atalanta.
That's max.
Yeah, go in again for me. I'm just still trying to.
When you're the Atalanta pilot
There's
or the Argus
Urchin.
pilot, your
yeah.
Oh, where?
planar movement in the XY plane is dictated by the movements of the Nautilus, which is what the nav seat does. So my controls are the winch, which controls Z offsetter, the altitude of the vehicle, as well as
Finally.
the cameras,
Never gotten a stable shot of these guys.
and then
Sorry.
the yaw, or the rotation.
They're always in a difficult spot.
If you're flying Hercules, it's more like flying a plane, and you've got, pitch, You've
Still
got
not
X,
this
Y,
shot.
you've got Z, you've
Can you
got
come down,
rotations.
come down, come down, come down, 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 down.
Down, down, down. I got 10 meters below.
Yeah, come down a couple meters. Just for the shot.
You just don't want the tug.
So then which one is, one, easier to fly, and two, more fun to fly?
Yeah,
Right
thank you.
Okay, moving
Coming up. Atalanta's certainly easier to fly than Hercules. I'd imagine that Herc would be more fun to fly, but you're going to have to ask Dan about that.
Dan?
Yeah, Herc's a lot of fun to fly. Learning is about like learning to drive a car. That's the easy part of this job for the most part.
The fun thing about Atalanta is you get to, as I'm not really doing very successfully at the moment, try and get nice shots of Hercules. And you also get to control some of the hydraulic functions on Hercules, like the sampling trays and the tool trays, some of the camera angles. And as I've been practicing, getting some time on the craft manipulator.
And you
So-
got a rock.
I got a rock. It's still lots of fun.
Yeah, sorry, I lagged there for that shot of that guy. I should go on the highlights. So
Sorry, I missed the
Still okay.
conversation about that thing, the red thing with like the spider webby things. Is that related to a sea dandelion?
Yes.
Yes. Okay.
I want to see
Looked
that one
similar.
on the website.
And Steve, what was that creature again?
Dandelion siphonophore, that one.
Siphonophore.
Oh,
Yep.
has it right in the name. Okay.
Siphonophores are generally the most frightening creature to ROV pilots.
The genus name is...
Why is that?
Just because they're delicacy?
No, because they
I
look
believe
like
it's
a piece of line.
Thermopalia.
Oh.
Thermo.
Yeah.
Interesting.
I think it's... I don't know if it refers to the color, like the red color,
Right.
or if it refers to the area where they were initially
Okay, Lynette,
encountered,
I can start
which were
moving
around
close enough to the
vents.
other side here.
They can be quite abundant around hydrothermal vents and warmer environments. But we find them everywhere. Seamounts are very popular spots.
Ooh, another sea cucumber.
Another black coral, also possibly Heteropathes, or that might be Lillipathes too.
I'm not going to stop for Zoom,
Yeah,
Steve, unless you ask.
I'm just making notes for
Okay.
data logging purposes.
Roger. Anyone
No
wants a
pennies in the Zoom
shout out?
bank.
No pennies in the Zoom bank at the moment.
Victorgorgia.
Thought we went with watchbox.
So here's another question from chat. What percentage of the science or analysis is happening while actively diving versus after diving?
That's a cool question.
I don't know if we really have any analysis ongoing during the dive. It's more of just data annotation and making sure that we catalog a time series of what we're seeing on the seafloor. There might be some impressions that we generate. Well, this site is similar in this respect to this other site, but it differs in this other way. Really simple initial notes.
And are these white, frilly, lacy-looking things the sponges that we were seeing earlier? Or
These are Paragorgia. This is actually, can we zoom in partial?
Sure, go ahead, Dan.
While we're moving. Yeah, these are Paragorgia sea fans. This is what we sampled a while back. Yeah.
Okay, can go in. I think you just wanted to get the ID in there.
Yeah.
Marilla, Bathypathes, Stauropathes,
Saw some
tons and tons of bases here. So you can tell this has been a pretty prolific community over time. A lot of old Paragorgia bases, bamboo corals, maybe 2 or 3 different species here.
Coming tight.
Are those the larger heads or colonies in the background? Oh,
Yeah,
there it is.
yeah, that's right. Yep.
Cool crevice.
Oh, it's a large Farreidae.
Well, this isn't as flat as it says on the Bathy.
Yeah, for real.
Pretty
I was
wrong,
thinking.
in fact.
Can you come back around?
Oh, actually, can you come? Yeah.
I cannot.
You cannot?
Correct, I cannot.
Can you turn 180? Are you able to?
No.
I'll turn auto head off if you want to come around.
Yeah, stop the ship.
Okay, I'm gonna come back around here.
Yeah, I need to do.
Steve, here's a question for the watch lead. If you came upon a shipwreck, would you explore it as part of the expedition or come back on a different expedition?
That's above my pay grade, I think.
So what do you want to see, Steve? We're looking
Yeah,
back
so
the other way there.
off to the left, there was something a ways. Yeah, we're not out here to find shipwrecks. We're really interested in characterizing the seamounts. It's way over here.
You'll have to.
And even still, we're in a monument, so there are
We're about to come tight again here.
You can come down.
rules
5
that govern
meters.
those
Coming
types
down.
of
Couple meters.
archaeological history.
There we go.
Yeah, can we look back here at this sea star? Is that predation going on?
I see what you're after.
I'm gonna have to come up a little bit.
Good.
Roger.
Auto heads off.
Then to answer that question, how frequently do y'all discover these species? We do about once an expedition, something like that. But it takes a little bit to actually decide if they're a new species or not, because we have to send them to our labs on shore.
If it's once an expedition,
Go tail to tail if you're not already.
this is
That-
24
Yeah, we are.
hours.
Yeah, it's a chance.
That's a lot of hours.
Got you in my butt cam there.
Yeah,
Like
I wouldn't
500 hours.
I wouldn't necessarily put a hard number on it. It's likely that we probably see them, but sometimes it can take years or decades to describe species in whole. And oftentimes it's not just one specimen that is required for a new
The ship's
species.
pulling
It
me
oftentimes
220.
requires
I'm
many
gonna
different collections
pull
from a
on you
very
here.
large geographic area. Sometimes, or sometimes it could just be many specimens from a smaller area.
Was
The
it
best
here,
species
Steve?
descriptions usually have
Have you seen
multiple.
it?
I think we're on top of it, actually. Are you talking about the coral?
Yeah.
Yeah, I think it's on top of us or just behind us, or under us.
Okay, I'm gonna try and come around now.
Nope.
Okay.
So it's a process. Collections we make might contribute to a species description,
I'm not seeing
but
it.
it might
no.
not be the first species. Generally, I think there's a hierarchy. The first collection of that, whatever new candidate species is usually the
Watch your rear
type
there.
specimen, which is a special designation, where the other one's called paratypes usually.
We'll have to move the ship, Steve. I can't get there.
Yeah, I think we're like right on top of it, just outside of the lower
Yeah.
frame.
Is it worth a ship's move to backtrack?
It's something I want to look at.
Okay.
Yeah, so
Okay.
there's 2 of them here. One of them is here.
Yeah.
Move to the northeast there. Just move this one that way. Okay, you can spin around now.
Coming around.
And then looking at so there's that one, and I think there's also one right here I wanted to look at too.
Roger.
So 2 things,
Oh.
2 possible predation events.
Maybe not yet.
And to answer that question in the chat, do you ever collect live specimens other than coral? So we
I'm
do
gonna give it a
a
minute.
lot by accident.
I'll come back. a little
Sometimes they'll
more. I
collect like
drug you quite a ways.
a squat lobster or something that's attached to a rock or
What's that?
that falls into the
Yeah.
ROV.
And I also think we also collect
There's
sea
a way to put it
And
next
sea
to
cucumbers, yeah.
Yeah.
All right,
Urchin,
turn around.
right?
Yeah.
Urchins.
Urchins. Did
Tunicates.
we collect that slime star? Or did we just leave it?
No,
be?
we
I
left
think
it.
we left
Okay.
it, yeah.
Here's a question for our ROV pilots whenever they're ready.
I forget how he does it.
How do you account for the current drift between Nautilus and the ROVs? Does that ever happen?
Well, I have this really nice feature called auto heading. Auto heading. So if I give it a set point, say it's 90 degrees, the thrusters are going to work to try and maintain that despite the current.
So it's still pulling you around?
Yeah, it's pulling me around still. Look at your butt there. It's
Oh, yeah.
there we go. I got you in sight now.
Gotcha.
Up and above.
So I pulled Atalanta quite a ways. 20 meters I was pulling Atalanta. That's why I couldn't turn it all, because I was dragging Atalanta with me.
But the current does pull Atalanta away from the stern of Nautilus. And from what I've heard, because it's lighter than Argus, it can be a little harder to predict as well. It's just a matter of keeping an eye on their position from the USBO and sort of anticipating your movements to make sure you're safe and where you want to be. Nav plays a big role in that, making sure the ship movements line up with the scientific targets, which is what we're trying to do right now.
I don't see her moving.
What?
You asked her to backtrack along the same line, did you?
So Steve, it seems
Roger.
like we're seeing a lot more life, or a lot more lush life at this point of the ocean floor. Would that be correct in that sort of assumption?
Can you adjust the iris a bit there?
Yeah, there's some pretty good characteristics for this site. These boulders are really
Maybe a
great,
little darker,
stable
or?
substrate.
Oh,
They probably have some topographically induced flows, in addition to some pretty strong
flare from the light.
currents from our deep current models for this area.
You want auto heading off?
So that kind of is the perfect storm
still
of
pulling you?
Yeah, I'm turned around.
Roger. I'll come
environmental
back again.
characteristics that would support this kind of community.
So then do we know what's making this area a better candidate for life, for like in gardens and colonies? Somebody in the chat asks.
Yeah, faster currents probably
I'm going to come back and come on
you know,
again.
for feeding purposes, but there's also probably a good larval supply from a lot of the species around here. There's probably a lot of localized recruitment or young corals and sponges settling in an area. We saw that with some of the dead bases on the wall of this boulder. So things have been growing here for a long time.
Oh.
Hey, Kukui.
Yeah.
What sample is 075?
075.
There we are.
That is a coral colony snip.
Okay,
Yeah.
thank you.
Yeah. Thank you.
So speaking of the coral colony, somebody in chat wants to know: is there a risk of barometric damage to corals when we collect them?
No.
Simple as that.
Could that be because of their sclerotides, unlike sea cucumbers, who just become glob soup when they come up?
I mean, yeah, there's no real barotrauma experienced by corals. They really don't have any air-filled sinuses in their body, like fishes or something
Take
like
that
that.
wrap out.
So there wouldn't be any
Yeah, I'm
trauma,
gonna take it
at
out
least
right now.
superficially. Cellularly, that might be another story, but
It's not something that really will make too much of a difference. Oftentimes we'll bring an animal up and it'll function just fine at the surface. Corals will often extend their polyps if in sufficiently cold water. And they usually can be kept for a couple of days even on deck, on the ship in the fridge.
Wow.
So, yeah.
Okay, you can come up a bit now.
Coming up.
Some more bamboo corals here.
Yep. I've lost my bearings here. I don't know where
We're like 40 meters away.
Okay.
You want to get back there, right?
You want to get back there? It's probably. You're gonna have to keep her moving.
Atalanta heading 045.
There's a cool view, Gibson.
That's a cool view.
Nice.
Get that darn tether out of the way.
Some cool view of Herc slamming into the cliff there.
You want to try a zoom?
Yeah, sure. It's cool just to see that thruster.
Freaks me out because I feel like I'm closer than I should be, you know?
Oh, yeah, yeah.
The Niskin's ready for water. To be closed, I suppose.
So Gibson, do you have like a sort of like cushion zone of how far away you should be from Hercules?
I'm giving myself about 10 meters of altitude minimum, whether it's Hercules or the bottom, but more comfortable is 15. Depends on the heave of or the pitch of the vessel, because I'm tied to that winch. So if it's nice and calm, 10 meters feels good. On instruction from my pilot Dan here.
Yeah, if we're touching, we're too close.
We're touching, we're too close. There you go. Right now I'm 18 meters above bottom. That's probably about 10 to 15 above Hercules.
Zoom in just a touch if you want to clean up the bottom next. Maybe out just till you
Good diversity on
to
the
see
rock.
it.
See some Bathypathes, Stauropathes,
Yep.
Crascorgias.
Hemicorallium.
It's going to be
These,
a leisurely trip
nodal
back.
branching keratoisidids.
So could we classify this as a coral
Unbranched
garden?
keratoisidid. Yeah, I think you probably could fit that definition. But I don't, yeah, I always thought there was a spatial component, you know, not just the density. Sometimes, you know, having a very densely populated boulder could, you know, fit the definition, but I think it's also more about the length. In some ways about the length
Bring
of
the PSC
persistence
back up when
and
you get a
area
chance.
of that density. At least in the original description, there
When
were
you get a chance, can you
fairly
bring the
extensive
DSC back up?
patches of high density.
What's that?
Gibson, you were saying earlier it's is a USBL?
Sorry. Pardon me, Dan?
You were saying earlier an acronym, was it USBL?
Ultra short baseline, I think is what it stands for.
Oh,
It gives us our position fixes of the two vehicles on the surface. It's acoustic location.
Gotcha. Thank you.
I think that, I don't know, Lynette or Rennie would be the people to ask about inner workings of USBL.
Mm-hmm. Cool. Thank you.
Yeah, you're welcome. I still got lots to learn there too.
Pure digital magic nowadays.
Pure digital magic.
Yeah. Proprietary digital magic. Our Ranger 2 head is awesome. It's got its own gyro in it.
In the left-hand corner?
Left side, yeah.
And a Farrediae right in front of us?
Yeah, that one looks like it's dead. Not much going on there.
So Steve, someone in the chat wants to
Wet
know,
newspaper.
do we ever find like non-organic things at this depth, aka like man-made things?
Yeah. Marine debris. Often.
Just gonna do some checks, Dan.
Well, can you
Sandals.
zoom in on this for a second?
Sure. I'll need that
Looks like Stauropathes.
bottom camera there for make sure I don't plow any of this stuff. Okay, go ahead, Dan.
Gotcha.
Can we set up for maybe a collection of that?
Sure.
On our way to the other collection?
Get out without knocking this.
I'll let you know if we need to abort, but I wanted to let you know earlier than later.
Very fragile.
Falls like a wet
Yeah,
newspaper.
we're headed that way, Steve. We've moved the ship, what, 30 meters, and Atalanta's moved 20.
Yep.
And we've moved 40.
It's worth lingering a little bit.
Yeah, totally.
Yeah.
There was some really cool stuff there. We were sorry about the exit there. It was when I pull Atalanta like that, it rubber bands me back, and I couldn't stay low backwards there. I would have just plowed through the coral field.
So this might be a different Stauropathes species. You can see the two here, the mustard one, and then the ketchup.
Oh.
This pinkish red one. This might be a different species.
I can let her stop after this
Put
move.
mustard and ketchup on my pizza.
Yeah.
Ooh.
No, just let her finish the move,
Interesting.
but we'll hold off before we do another one.
Curious why you have two color morphs here next to each other, though.
Why do you think that is?
I don't know.
Same species, different colors? Is that what you're saying?
Possibly the
Competition.
same, possibly different.
I have a lot of heading on, that's why it's...
And the yellow branching one by the yellow morph?
It's definitely got a very, a slightly different branching sequence.
Atalanta heading 000, standing by for use of the bubble cam.
And is the yellow branching one also a Stauropathes too? The yellow mustard branching
Yeah, that's
one?
Stauropathes, yeah.
And the one in front of it? The fluffy
Oh,
looking
this one? Which one?
By the ketchup looking one up to the north and behind it as well.
Yeah.
Got the DSC on it.
Stauropathes.
Imagine that. Okay, Dan, you can also zoom in.
Sure.
We don't need,
I take the bubble.
we wouldn't need much of this.
You can full zoom if you want.
Sure.
You know, maybe something like that.
Oh, wow.
It's a very interesting pattern that it has.
Great shot, yeah.
Yeah, interesting polyp.
Totally
formation.
different branching pattern than the yellow morph. So this is a worthwhile collection. I know the yellow has been collected in this, if not in this area, then elsewhere in the region. It's fairly widespread, but this one I'm not familiar with.
Come up nice and easy.
So it could be a
don't pull
different
me off.
Stauropathes species.
So when we collect these type of samples, are we all also testing for stuff like
All right,
bacteria?
we're all
or
set back
archaea?
here. We can commence collection.
Yeah, I'm just waiting for manipulator telemetry. We have a, since we have a ground fault on the thing, we're turning the power to it off and our ground fault. Then when we turn the power on, we're going to turn our ground fault sense off. Otherwise it goes crazy. So it takes a little longer to get it out than usual. tried to stab me in the face once, so I'm a bit nervous with it. Okay, open the, chase me with the bubble cam there. Yeah, that's what I want to see more of the, just show me the tip of the minute. Okay, show me the victim there with the bubble cam.
Come again?
Show me the victim with the bubble cam. Sorry, the sample.
What's our depth here? 1800.
That's it there.
Can hear that music again.
Dun, 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 dun.
I have to do it one more time just because I can. I'm actually watching it in the other camera. It seems more interesting for you guys to be able to have the tight shot than looking at a manipulator. That's my rationalization there.
We can go, I think we can go into this bin also. We'll just start dumping all the stuff in there.
Lambda?
Yeah.
Okay. Sounds good.
So, how
That'll
much
be
of
our
this guy do you want, Steve?
Yeah, yeah. Well, let me draw it for you.
Right
Yeah, this is plenty. We don't need much.
Roger.
Yeah.
So going back to that question from the chat, do we ever test for other organisms such as bacteria or archaea? If so, how?
Not on this expedition, but if you were following along on the last one, a couple of months ago, there were some microbiology research going on, specifically relating to the crusts that form on the rocks here, by Dr. Beth Orcutt, who was working on, all kinds of different microbes that live in and around and help facilitate, or possibly help facilitate rock alterations or crust.
Where do you want this guy, Steve?
We're going to go in the forward bio box on the side with the rest of the corals, the left side.
Copy that.
Lambda.
You can turn the
Hope that
torch
naming
light on there
convention
when you get
come
a
about.
chance.
Yep.
It's not already on.
How's that?
Perfect.
Yeah, lambda.
Oh, sorry.
Zero 7 8. Coral.
Data, is that 078?
Correct.
Thank you.
I think it might be lowercase.
Well, what I'm doing here is
lowercase.
getting the shoulder gets cut up if I come in too high.
Okay.
Okay, you can should have room to push it open there. Keep an eye on the DSC there.
But uppercase omega. That's
Don't you float. Okay,
all right.
you can close it.
give you a DVL reset?
Sure. Want to zoom in on the other guy back there? Or are you going to want to sample of him too? Or
No, we can skip that one. That one has been sampled more widely, I believe. Good imagery, though. All right.
okay, you can start moving the ship again. Or maybe I can get,
Yeah.
let me see
Good.
where.
Try to get there from here.
Yeah, let me see if we can get there from here. Can you turn off the porch light too for me?
To answer that question in the chat, if there's a schedule available somewhere, I don't believe so, but every watch will give you a brief summary of what we're doing as we do a watch change or every so often.
If you're loving us, come back
4 to 8.
4 to 8 Hawaiian time.
Aye.
That's our little time to shine.
Except we'll probably be on deck.
Right. Well, next time.
Next time. Tomorrow.
4 to 8 to
Tomorrow
join
night.
Delta Dan and the
Well, let's see. No, probably.
Delta Dan and the Benthic storm.
Benthic storm.
Coming at you 4 to 8.
There
Like and subscribe.
Live.
Too bad I have 0 musical talent.
It's okay.
You can get a
Play the
radio
kazoo.
DJ
That's common
voice.
for lead singers.
You
Yeah.
can sing.
Yeah, frontman
No.
Dan.
I've been told I can't sing. I'm allowed to set up the lights and plug in the amplifiers. That's it.
Getting back to that starfish, hopefully.
Yeah, we're going to see what we can find, if we can acquire it, and take a look at what's in the area. Maybe we can do one of our planned triad samples, and then I could be
You
happy
can,
and sleep well that we've got at least one.
You should be able to spin around.
You sleep, Steve?
Occasionally.
We just prop him up right in the closet.
Yes. I don't know why people keep saying that. I think I sleep a lot.
045.
What's that?
I'm
It's
having
not, it's not the
045.
first cruise I've been on that people say I don't sleep, because I definitely do.
It's such a strange feeling, right? That you're like, okay, I'm sleeping. Am I sleeping the right amount? Am I sleeping too much? Do people need me? Am I awake?
Actually, during the blue water this morning, I was definitely on autopilot, probably regenerating 50%.
Go to cruise control mode.
Yep.
Non-sleep deep
That pizza
rest.
talk pumped your adrenaline
I just
up.
have a series of pre-recorded answers.
Bamboo coral.
It's not the first time I've gotten these questions.
I challenge our viewers in the chat to come up with something that Steve's never heard before.
Don't do that, please.
See that tether there?
Yeah, I'm seeing if I can tie it in a knot. Looks like I've succeeded.
Nailed it.
That's a cool looking channel.
Yeah, this is north-south oriented little canyon here.
So we're right back near where we saw the target.
Yeah, it's just to my
I'm
left
gonna
there.
guess over here, Paragorgia.
It's over on this rock over here, I believe.
Oh, there's one.
Yeah, I think that was him.
Maybe looks thick.
Yeah, we can look at that one.
Sure.
So Steve, tell us why we're interested in this particular one.
So we're doing some exploratory some exploration of kind of predator-prey dynamics in the deep sea, and we're interested in observations of predation and how corals might respond to those types of predation events. This sea star here seems to have eaten several colonies in this area. I'll finish that answer after I take a look at what's going on here, because I want to make sure I'm making the right calls. We need to find a couple different individuals of the same colony in this area. And a lot of them seem to be predated, grazed.
Okay, Dan.
So the goal is we're looking for one that's
this is
like this being eaten and one not being eaten. Is that right?
Yeah, so this is definitely eating on something. Predating upon this, I think this is Hipposterius, the sea star, preying upon a bamboo coral here, internodal brancher. Okay, can you zoom out a little bit? And pan to the right. There's a colony to the right that I want to see if it's the same species or not, and if it is.
Zoom right out. Zoom out, please.
Zoom out, sorry.
Then we can go ahead with this collection, but I wanted to see if it's been at all grazed, because we also need a good example of an ungrazed colony.
I'm actually panned all the way around.
This way.
Maybe I can see it.
It might be off camera.
Yeah, I'll have to move a little bit.
Right behind the arm.
Yeah, figures.
It's sort of lumpiness on the stems, probably the wrong word. Is that from it being predated, or is that kind of what it looks like?
Can we do a zoom here?
Sure.
I just want to see if they're the same coral or not. I'm thinking they're not, in which case might need to look around a little bit more.
Were you seeing the rounded lumpiness, Lynette?
Yeah.
Like the pink ones?
Yeah, the one that the sea star is on?
So this is a internodal or just above the node brancher.
It's full zoom.
I don't think it's the same species, but could you...
You want to look back at the sea star one?
Yeah.
Yeah.
I think they're different.
Yeah.
This one's, different. Okay, there was another predation instance. I'm going to say no to this one. There's another predation instance. I think it was right behind here somewhere.
Roger.
Right, there were two sea star eating events. I think it was somewhere else around here. If my bearings are right, I think it was somewhere in this pocket.
Yeah, let me come up and get an overview here.
They kind of look like the polyps closed up to me, Lynette, like it had, sucked its polyps back in.
Yeah, it kind of looks like they're like super sucked in, or like maybe just the tops of them have been eaten off.
Can you zoom in on this?
Yeah.
On the big one there?
Yeah, the big one.
Yeah, go
just
ahead, Dan.
From here is fine. Yeah, okay, that one's...
There we go.
That one is also different. Okay, so 3 different bamboos, this one, okay. All set there. 3 different bamboo species, and also I think this one is a nodal brancher, so 3 different bamboos right in this area, not helpful. We need, for this particular experiment, we need 2 of the same species next to each other. One that has been grazed or is being grazed, and then one that is
zoom in just a touch
unimpacted.
to clean up the bottom there. Thanks. Tilt it down more than usual.
And like for example, this coral and this coral are probably the same species, but this one is completely naked, has no tissue left, and this one's in the process of being eaten.
So this kind of sea star here that we see, just kind of chilling in the coral. Someone in the chat wants to know, what's the life expectancy of these kind of guys?
The sea stars?
Yeah.
I don't know. I would wager it's probably a very long time. I don't know if there's an easy way to date them. Either, radiometrically or something. Not sure.
That's probably like 1.5 meter, 2 meter tall colony right there.
There it is.
Yeah. Yes, this is the one.
Cliffhanger. I love this one.
Good eye.
So, are there any other species that look like this in the area?
How big is your area?
On to the right.
On this rock or on the next rock. Can we see what the branching pattern is like this one?
Go ahead, Dan. Just zoom in there.
You know what? This one is also different too. This
oh my gosh.
This bamboo has massive intertentacular sclerites here, like that come out between the polyps, which is a totally different polyp morphology than the last ones we saw. So this is not helpful.
So interesting that like the stomach isn't out on this sea star.
Yeah.
It's just clinging with its 2 feet.
Can we zoom out and see if there's anything resembling this coral in the area, maybe over here, one of these colonies?
They all look the same.
It might have just been brittle stars, but that big colony on the left had something red in it.
So Steve, why are we trying to find our multiple of the same kind of coral?
So we're trying to do a comparison between some genetic characteristics called transcriptomes of one coral species versus another, one that's undergoing predation and one that is unimpacted. We're not collecting a lot of data. Yeah, this one's different also. It's a nodal brancher.
Okay, go away.
Okay, well.
Sort of when we were looking at that star, there was one that looked similar, kind of on the bottom right of the screen.
Try it. Let me back up a bit here. I got to come up too. As you can see with my starboard camera.
Yeah.
So, we can take a look at those.
That one that looks a little tipped over, kind of center bottom.
This one? No.
No,
yeah.
That one, yeah.
Yeah, that one's on the bottom. It doesn't look very good anyway.
Okay.
Kind of come around the rock here. There's a bunch of them we can't see, I think.
I don't want to waste too much more time, so.
We're here. I can just come around the rock and look from the other side. You might see something. Get up above these big guys and spin around here. I'll be able to get down closer. Can you come down?
Can we look at that?
Down
5 meters.
Yep.
At that big one that's right underneath our tail right now.
Yeah, can do.
It was on the adjacent rock. Yeah, this one. Can we take a look at that one close up?
Not much tether left.
Yeah, you should be good to come down another 5
coming
meters.
down.
You want to zoom on him or you?
Get any tighter?
Yep.
Yeah. You want to see the branches there.
Yeah. Polyp morphology looks different on these as well. And it's not a nodal brancher, it's slightly above the node branching.
So this is the other side of that rock there.
Is that another star? Starfish on the...
No, it's
the
just
same one.
Oh.
star
Yeah, if you
from
see
the,
any other stars, let me
Okay.
know.
star from the,
Different angle.
yeah, I spun around to, I can, to the left here, there's more guys on the same rock here. Maybe.
It's in the middle of the screen there. Middle bottom.
All right. Well, can we just take a last look over here?
Sure. How close do they have to be?
Um,
It's only a couple meters, right?
just wanted to see some details of the colony. These colonies over here.
So these colonies look oddly like blown over. Is that because of the water column, or?
Not sure.
Yeah, really interesting.
One zoom in. If these aren't them, then well, we can
Go ahead, Dan.
pick up and move on because
Real star's fine.
Oh, nope.
No.
This is
no
the one with
cigarette.
like the fleshy, like the thick fleshy.
Yeah.
Sometimes
Okay.
what they're called are volcano polyps. Okay, forget it. Let's keep moving upslope in the direction that we were going to waypoint 3.
Skunked.
So what were those little? There's like little orangey little dollops across each of the stems. Were those parasites or?
I didn't catch them.
Oh, no,
So
They
they're
weren't the polyps, right?
Maybe? Anemones, maybe?
Let me come back
They might have been
around.
anemones, yeah.
That's a cool shot of Herc.
Yeah, those.
These guys.
Yeah,
Those are cool.
anemones,
Yeah,
yeah.
those do.
Bamboo corals are hard enough, which is in part why we like, for this experiment, or we prefer primnoids, because it's more easy to tell, you know, two of a like species. But with bamboos, there are many other morphological characteristics you have to gather to tell if they're the same or different. And sometimes it's hard, and the experiment doesn't work if you don't collect the same species.
Ah.
Still a whole bunch here on the, this is kind of where we came in, all these guys.
That's crazy
Does
crochet.
it count if they're down on the rock, does it?
Okay, you bring it back up.
Yeah, that's awesome.
Nice
Yeah.
zoom.
Yeah, I didn't see any when we came in this way. This is the way we came in.
Right.
Any sea stars or predators?
Okay.
Yeah, we could
was-
spend all day looking around here, but I think we could just as easily see a lot more up towards waypoint 3. We have another
Yep. You can,
cone to climb up.
you can kick her into gear, back on the path. Tilt
This is
down a
great.
little
I
for
would
me.
love to scuba dive in this kind of landscape.
It's beautiful,
That'd be
right?
amazing.
Maybe not at this depth, but
Yeah.
Be wicked if we saw a dike running through this basalt.
Very cool.
oh,
That'd be awesome.
Wow.
What would you
Look
be
at that
excited about,
amazing
Brandon?
colony.
A dike. It's this volcanic feature where a new tube opens up and it penetrates through the old rock.
Ah.
And it looks really cool because it doesn't look like the surrounding rock, and it usually is oriented completely opposite or way different. It basically just looked like a 2-meter long beam across the entire rock of a different color, most likely, if it was young enough.
These are so gorgeous.
These are amazing.
Which are these, Steve?
Oh, most of these are bamboo corals of one type or another. I think these look like they're possibly nodule branchers. Maybe J-clades, but I'm not sure.
They're
So,
amazing.
Steve, in this experiment, somebody in the chat wants to know, are you looking for
How tall that one
apple
is.
cards?
Aplecopherans?
Yes, that. As well as part of the coral predatory study?
If it was the right colony, I would consider aplecopherans, but it's not ideal for what we're trying to get at. We need more tissue than what an aplecopheran can provide. So yeah, we're looking for sea stars or sea urchins preferentially. So the idea behind this experiment is trying to see what kinds of activities corals are involved in during predation. You know, what are they doing at a genetic level? What are they doing at a genetic level? There's a sea star over there to the right. To help compensate for the fact that they're being eaten alive. Do they engage in wound repair, for example? Might have some luck here.
Zoom in on that guy real quick.
Another lounger.
I just love these things. So they're just folded over.
He's like on a hammock.
Just chilling. If your hammock was made out of like hamburgers. Yeah.
Living their best life.
It's an all-you-can-eat hammock.
Yeah.
So, Steve, what's the next step once you find what you're looking for?
Just a second. I'm gonna.
Another one.
All right. So this is like the fleshy one, nodal brancher. Is there another coral in this area that looks like that? Can you zoom out and see if there's another individual colony that looks like this?
It looks like it's relaxed, but all the tube
That
feet
one right
extended. It was
Zoom in
reaching
there.
out.
Yeah,
trying
can
to
we
hold
zoom
on.
in on
He's stretching.
that one
Mm-hmm.
Just took a nap.
on the left there?
I'm so excited.
It's Max.
Drum roll.
You're killing us with the suspense.
I
Steve.
know.
I can't tell. I can't tell.
We can get a little tighter.
Yeah, zoom out and look a little bit more broadly. I think those two are actually different.
Yeah. This guy here. That's gotta be it.
No, that's that one.
Yeah.
Oh.
Taking a still and comparing them,
No.
are you, or you just?
I don't think so. No, I'm gonna pass on that one.
Roger.
You're looking for are they the volcano polyps? Is that?
No, these ones are.
Is that like a little hole in the rock? Like a
They're
little cavern?
longer polyps.
The bubble?
Oh yeah.
All right, let's pass on this one and keep going.
Some more to the right there.
Oh yeah, this is this one.
Wow, it just keeps going.
Yeah.
There's a lot of bamboo corals.
Just a couple.
Love it.
We're here. Might as well look.
We may as well do it.
Take a peek here.
Go ahead then.
So we're looking for the fleshy ones, right? Ooh.
This is why I don't like bamboo corals.
Look so similar.
No, this one.
Why do you have so many different morphs?
I really don't like bamboos. They're very confusing.
That was bad. When you were a coral on Steve Oscovich's Don't Like list, you've done something bad.
Let's pass and just keep going on this one. I'd rather have prinoids anyway.
Yeah.
Suckers. I'd rather have something else.
Sick burn.
Anyway.
Have a look at the sea star on the way out. No, you can move the boat. You can do one more zoom there.
Sure, yeah.
It's kind of a cool shot.
Check out that star.
That's good, yeah.
Looking for that?
All right. Well, it was a good shot.
Yeah.
Is that a brittle star on a sea star?
There's
Yes,
probably
sir.
a brittle star on there. I'm not sure what the brittle star is doing.
Maybe they're friends. BFFs.
Very, very Disney perspective on nature.
Sure,
Thank
they're
you.
friends.
They're hugging.
I mean, why not?
Yep. And maybe I'm a
We
Chinese
have nothing to
jet
prove you
pilot.
wrong.
The unfortunately, yeah, echinoderms, eating echinoderms are some of the more common deep sea
Oh, no.
predation relationships we see. Crinoids, everyone eats crinoids. Sea stars, sea urchins.
Yikes.
Brittle stars, I don't know.
Do crinoids eat other crinoids?
No, and I've never seen that. No,
So not everyone eats crinoids.
incompatible feeding modes to cannibalize. Yeah.
Is it actually? That's an interesting question. Do other carnivorous, any other carnivorous species, can they eat each other? Like, have you seen that happen on the ocean floor?
No, I don't think I've ever seen that.
Do you think it's a possibility of happening, or does it kind of like the unknown rules of nature?
It doesn't make a lot of sense evolutionarily. Yeah, because you know, there may be difficulties to find mates in the deep sea if densities are very low. So I don't think it would make a lot of sense evolutionarily for that to occur. But it doesn't mean it can't.
Anything's possible down here.
Even brittle stars becoming friends with the sea star.
Yeah, this like while we're, you think it would be possible while we're cruising to pop another Niskin bottle
Yeah, sure.
right here or in this area?
Sure.
It'll be a useful comparison to the last one we did as well, which was it 77? Was our last
You
one?
want to
For the Niskin bottle? Or for the sample?
turn
What was
the
our last
bender
Niskin
off
number?
74.
74.
Okay, so it's been a
power
while. Yeah,
only
let's
on?
do it here too. Similar
bender is
species,
off.
but
Yeah, now go to
yeah, so there's
hydraulics
similar
and turn
species
crop
here,
power on,
but
not the
we're
valve.
about 100 meters shallower and about half a kilometer
power's
distant.
on.
It would be interesting to compare these two profiles.
And give me number 5 on the bubble camera there.
How many species would you say is on here? 10?
This is probably between 20 and 30 in this area.
Oh wow. What's the most dense sort of, or densely populated, actually let me rephrase that. How many species have you seen clustered together, like the record that you've seen so far?
Thank you.
I've seen more dense communities than this. Hemicorallium gardens up in where were we? Last year, Voyager seamounts were pretty incredible. I don't think I've ever seen anything denser than that.
Can you give me the porch light?
Voyager. Where are
Porch
those
light
at?
coming
Steve,
on.
have you seen the
That's due north of here, I think.
have you seen the oceanography supplement?
I did skim through it, yeah.
The cover of it is one of
from
those
that cruise.
photos, and it's
Yep.
awesome.
Yeah.
It's just like chaos of corals all together.
Yeah, that was an impressive.
Niskin pulled.
We had such a great dive that we saw, we had planned to do many other seamounts, but we had to dive that site again, and we just found even more dense communities. I think it was Tamana seamount.
Was that Niskin bottle number 3?
Yeah, it was.
Awesome, thank you.
Yeah, Tamana seamount, I believe, was the face-melting hemicorallian gardens.
If you want to see highlights from that cruise, you can click on the expedition page and then head back to last year, 2021. It was expedition NA134, and its name was Lu'ua Ehike Kapapaku, one of the expeditions that we had named inside Papahanaumokuakea Marine National Monument. So check that out. Check out all the team profiles and a whole bunch of highlights and photo albums of those corals and sponges.
And these little bushes of coral, so someone in the chat wants to know, those smaller bushes that are to the right along the ridge of the rocks, are they chrysogorgias?
That's right.
Gonna see our lounging friend again?
I've lost my bearings here. I don't even know what direction we're pointing. I just see corals all over the place.
This is where the lounging guy was, yeah.
Oh wait, that's the same one?
Yeah, that's the same one.
Oh.
Because I remember the fracture in the rock on the right.
Yeah, we've been here before. So actually,
we're just going in circles.
Third time through here. It's just so much fun. Hit one on bubble for me. And we'll look at the other wall this time.
That's right.
That's what I was going to do. There's that giant fan on the left there. I didn't want to look at, and I didn't.
Do you need a minute to catch up?
Nope, you can
keep going.
Moving. Yeah.
Okay. Bridge nav. Can we move 50 meters 240, please? Thank you.
Beautiful sunset from the aft deck cam right now.
Yeah, golden hour.
Oh wow, look at that. If you turn your attention to satellite 3 in just a second, or channel 3 in just a second, you should be able to see it.
I don't know which cam it is, sorry. Oh.
Let's see here, there we go.
Yeah, that's
nice.
7:30 local time here. Watch change coming up at 8. This dive is scheduled to be back on deck at 4 a.m. So a lot more exploration yet to come.
On deck at 4, that's the word now?
That's the word. Mid-dinner game time decision to extend for 4 more hours.
Bird, bird, bird. Bird's the word.
Extend like those lounging sea stars.
Ah, yes. I love them.
Lounging, fishing. A type of fishing, I suppose. They're just extending their arms out, trying to capture what comes through. Oh, there's another species we haven't seen yet. That's an S-clade.
The green one?
The yellow, green, neon. Yeah.
Wow.
S1,
It looks like it's
S1
going really
clade.
fast, like all of its branches are like blown back.
We also saw really super high density communities of these S1 clades in the same cruise we mentioned before.
Okay, Steve, so that one branch has exposed skeleton and then none. Could like something have been eating it and fallen off?
Yeah.
Like, did the sea star tumble?
Well, you know, the strange thing is we've actually seen them, you know, what appears like, you know, they prey on, you know, some bit of tissue and then they climb back down. You know, we've seen them, you know, just leave patches, almost like they're farming.
Yeah.
You know, like they eat a little bit and then they wait for it to regrow and then, you know,
so they make
save it for later.
Food?
I don't know. I'm totally anthropomorphizing now, but that's what I would imagine they do.
You know what? That's okay.
But some of them are just gluttonous. Some of them will just eat the whole colony.
I like to imagine my brittle stars with little sun hats and overalls.
Oh, that's nice. I like that image.
Send us your sketches.
Send us your sketches. Hashtag Nautilus Live on all social
artists
media.
in chat, you have a chance to win some watch books.
We have a lot of amazing artists that like sketch and draw and paint along with us.
The patches
and
have
share
been
their
awesome.
art.
The stickers.
Oh yeah.
Oh yeah.
Plus our.
You got to give Jess that idea. She does a lot of that. That very kind of.
She does.
Thanks, Jess, if you're watching.
Jess should be sleeping. Napping.
I mean,
up for midnight
good night, Jess, if you're watching.
Yeah, it's awesome.
You know, the interesting thing is we lost a bunch of the prinoids that we were seeing a lot earlier. There's still Candidella gigantea around here, but it's mostly bamboo coral dominated now, bamboo and chrysogorgia. That might be meaningful, but I don't know what it means yet. I mean, visually, and in terms of community composition, these seamounts that we've been diving on the past few days, if you didn't know you were in Johnson Atoll and you were in Papua-Nomo-Kokea, you could probably imagine you were up there. Very similar, if not identical, species assemblages.
Mm-hmm.
Which makes a lot of sense. There's there's fairly short distances between seamounts, what we might call stepping stones between those monuments up, the monuments up north and this monument unit. There's Necker Ridge and the large guyots up in the north of Johnston that probably provide some continuity, that you might have some gene flow.
Yeah, Steve, can you talk a little bit more about that? Like in the shallow water, there's really high connectivity between Papua-Nomo-Kokea and the Johnston Atoll region.
Mm-hmm.
Sounds like we're seeing the same for the deep sea?
Well, the species are definitely continuous. I think there's a fairly continuous distribution. Whether or not they're actively, and to what frequency they're, larvae from one place might reach the other, that's unknown, and it's very difficult to do that type of analysis. Oftentimes to do those types of population genetic analyses, you need a lot of specimens, 20 or 30 specimens of the same species at a minimum from each location. So you're talking about a lot of community impact.
Yeah, that was cool.
But you could draw some similarities probably. Maybe you might be able to do some population genetic analysis with what's been collected so far, but I'm not sure how much strength there would be to it. There has been some work done on population dynamics in the genus Narella across the North Pacific, and my understanding of that study was that
See something interest you there?
was that there's really not enough known, or at least the genetic markers that were used didn't really provide any useful information. So that's the other kind of mechanical challenge, is that you need really good genetic markers that tell you something about small differences in the genetic material of these species to determine if the strength of connectivity between two sites or among sites. It's a very complicated question. Yeah.
Some of my first field work, not deep sea field work, but field work, was doing that type of parent and offspring generation connectivity work for tropical coral reef fishes, and we had to collect so many recruits, like tiny recruits of the year, and we were working in the Bahamas all around the Great Bahama Basin, and what that kind of like deep water to shallow reef community. And it was years and years and years of work before we found, but we found individual like parent and offspring connectivity, because we had marked adults, and then would track where we'd find recruits, but huge sample sizes, yeah.
Yeah.
Is that a large Paragorgia colony on the left?
Or is that Farreidae on the left?
I think so.
Kind of looks like
right
it.
here.
Yeah, you can see this spiral looking thing on it.
Farreidae, new species, currently being worked on. I don't know where the progress is at with that, but still.
That's awesome. Sorry, I just looked at the chat. Dr. Sako Matsumoto had the same question, and that I feel flattered to myself that I made the same ID as Sako. So for our viewers at home, just a refresher that there are folks all over the world watching, all types of fans of exploration, from professional scientists and experts in their field who are helping us identify species and contribute observations, and students, camps, museums, science centers, fans of exploration. We're happy you are all here. If you don't already follow us on all of our social channels and web channels, know that we broadcast here on Nautilus Live. If you're watching on YouTube, we broadcast on our website, Nautilus Live, and take questions there live into the control room. You can also follow along with the expedition on Facebook, on Twitter, where we send our dive alerts every time we go into the water, on Instagram, on YouTube, and on TikTok, and on LinkedIn. So anywhere you want to find us, we're really glad to have you along on the expedition.
Oh, see an enemy right there.
Am I pulling on you there?
Yep. I feel like we're lost in a maze of boulders and corals. Can't get out.
New band
I'll
name?
catch up shortly.
Boulders and Corals?
Boulders and corals.
And benthic storms?
I don't
It's
think
still better, I
I don't
think.
think anything beats still there the benthic storms. Although the swimming cryons
That's
is a
a
close
crazy
second.
base of that bamboo.
That's amazing.
It's 20 centimeters plus.
Oh, whoa.
Now, would you call this a thicket of bamboo coral?
There you go.
Yeah.
Put it in a paper. Write it up. One day
really
somebody will
concerning.
be talking about you on Nautilus Live.
Oh, jeez.
Yeah.
I'm there now. I'm good.
Dr.
Sorry,
Kukui
I'm
Gavana is here
pulling
in the
you there a
Scientist
bit.
Short Chat. Thrilled to have her with us.
All right, we have a candidate predation event here.
Oh, right in the middle.
Yay.
Defense.
Ah,
Of course,
yes.
is the hardest one.
And same as the last time, I'm pulling Atalanta.
Fortunately, I can tell you that the left and the right colony are the same.
What about the middle one?
Okay, let's
The
move.
middle one that's being predated
Yeah,
upon
we need
is
that
not.
here.
Ah.
So these three colonies are the same. 1, 2, 3.
Yeah. Need a.
Do you want to attempt a collection here, Dan? Do you? How do you
Yeah,
feel?
I'm at the moment. I'm pulling Gibson tail to tail, so we'll have to move the boat closer. Right.
Well, the good news is we can
Bless
only do one of these per dive approximately, so this will be it.
So I can look around on this heading while we move the boat. I can't really. We can like get some shots of the base here or something.
So with these collections, for sampling space, we're going to need some room. What do we have open now?
Get some DSCs there from this angle if you want. And you can zoom in there, Dan.
Sure.
Wow,
Zoom
look
right
at
in
that
on
thing.
base.
Yeah. Let's check it out.
So is that all one coral or many corals going after one base?
Wow.
That's probably one base that has been so heavily calcified over, it looks like it's fused. But that's one coral, and it's probably branched very low, and then the branches get overgrown by the calcium carbonate at the base. Yeah.
Look at it. That's
You
like,
won't be
it's
able
like
to zoom
30
too tight.
centimeters
I'll have
to
across.
go closer.
Yeah,
Yeah.
the pinkless
I'm,
lasers. Again, the lasers are 10 centimeters
Bobbing
apart,
around
about
for
the
a
width of your hand. So
should
just
be
thinking about
good for another couple meters.
2 or 3 palms
meters.
wide.
So this thing is huge.
It's huge. Oh yeah, at least 3 wide.
Wow.
Do we have open for box space? Like what's our sampling diagram look like?
Okay, you can go away.
Wow.
It's killing.
Because we're gonna need 3 bins.
This looks like an
We can do
enormous
C,
sagebrush.
D.
This
Double
is
up on one?
definitely a
I wouldn't want
coral
to put it in the
thicket.
bottom here because if they put rocks there and then yeah.
So we
So,
are
but we can
setting up
put,
trying
like,
to figure
bamboo
out
coral here,
this collection.
bamboo coral here.
Again,
And
this
it
is a
will
predator-prey
and then
experiment that
rocks,
is
yeah.
special to this expedition.
down
that
to the
we're
wire.
going to
Yeah,
collect
and that would be
the
close to
coral.
rocks.
Okay, so we can do
If we-
E,
Okay, the
F,
key
And
thing
then
we
maybe
spotted
one
here
of
first
these.
is that the
Yeah.
sea star is
Okay,
eating
one
the
of
The
these.
coral.
ship moving?
We're going to collect the sea
Yeah,
star,
she's moving.
yeah, it
She's
the
is.
coral that it's eating, and then its neighbor that is not actively
stuff.
being predated upon, so that we can look at the gene and specific like protein expression, figure out what genes are being turned on and turned off, maybe
That looked good.
defensive toxins, maybe regrowth, maybe reproduction, you know, just trying to understand within all the like mechanisms and systems
Is that
a
the mocha?
coral has to respond. What is it doing when it gets the signal that a predator is in its branches eating the polyps? But it's going to take a couple collections, so we'll be here for a minute.
Ah, that's loud. Sorry. The microphone muting when I told it to boom up. Is that as loud in your ears as it is in mine?
It was pretty loud.
Sorry, I shouldn't do that. It's a really bad switch. Still tail to tail there, as you
Yep.
can see in the
I know, it doesn't seem to have
Back
gotten any
down.
more slack.
So Steve, someone in the chat wants to know, have we seen many Walteria sponges yet?
A few, yep. We have seen them for a while. I think we've seen them most of the dive, in fact. Yeah. So we're going to need to access that sea star. You think you can do it from here?
it would probably come around and land on the rock there.
That might be better.
Yeah, for sure. Might be able to zoom in on him there though, if you want. Get some imagery here.
Is that an enemy? Is that a fly trap an enemy on top?
Oh.
No, I don't think it's a fly trap. It might be a different kind of anemone.
Oh yeah.
It could be, I don't know.
This
Maybe
is a
different
tank
species.
of a sea star.
Yeah, I was going to say.
Wow.
Fat, happy.
Fat
Got
with
a
P.
full belly of bamboo coral.
Yeah, you can see the like raw skeleton.
Path of destruction.
Yeah.
Like Godzilla
Yeah.
stomps through a city, you know?
So he's eating up all those bare branches below is all his work, is it?
Yeah, almost certainly.
Certainly seems good. So
Is there any way to tell, are sea stars hermaphroditic?
Oh, that's a good question. I don't know. Yes, I don't know.
Maybe a question for Liz. She's a echinoderm specialist.
One of the sea urchins we collected yesterday had gonadal material, so I don't know what was
That's
the
cool.
result of that.
Yeah, Liz was showing that many of the sea cucumbers are hermaphroditic because it's so difficult to find, you know, mates. It's a good strategy.
Are we waiting on the ship?
Yeah, we are.
Okay.
You see the tail to tail there?
Yeah.
Yeah, I can't do anything but what we're doing now.
So, Steve,
If
with
I let go of it, it rubber bands back.
with these sort of sea stars eating the coral, does the coral have any defense mechanism? Like someone in the chat wants to know if these corals are maybe venomous or poisonous.
I don't know if these have any of those compounds. Some corals do produce them. You know, we usually refer to them as like secondary metabolites, so they're, you know,
might have to
things that, you know, maybe unpalatable or maybe beneficial to the coral. You know, these are things that also humans have been known to use. They're sometimes termed natural products that are produced by corals that can be synthetically created by pharmaceuticals or imitated by pharmaceuticals. So, I mean, there may be mechanical deterrents like spines or something like that, but it doesn't seem like that's a problem for these sea stars. Oftentimes the sea stars are, you know, they don't really care what kinds of stuff is on here. Although it was interesting that yellow S1 clade we saw a while back, there was that bit at the bottom that was grazed, and then it stopped. And I'm wondering if maybe there was a grazer on there and it started eating something, and maybe there's something about the color morph that was impalatable. But that's just pure speculation right now.
So Steve, some of the detritus that comes down as nutrients, is that like all the way from the surface from like, you know, like, I don't know, a plankton bloom dying off or something like that?
But maybe the next one.
Most of the marine snow is probably a mixture of you know, dead bacteria and plankton from the surface ocean.
Yeah, because this is swinging
zooplankton,
right now. This is
skeletons, and shells, probably maybe some very fine-grained sediments in there. So it's a very gooey, mucusy.
Starting to move it that way.
Environment.
No.
Are there
let's see what happens here.
Seasonal patterns from the surface growth and decay that kind of reappear on the seafloor?
Yes, that's the idea. Typically, you get higher signals of seasonality in things like food pulses and marine snow pulses at higher latitudes, since seasons are typically stronger at higher latitudes. You have more well-defined seasonality, but at lower latitudes and in the tropics, it's not necessarily the case.
Can spin around now.
So it might be more of a muted signal here.
And is that also because of the levels of primary productivity between the tropics and the higher latitudes as well?
Right, yeah. Yeah, wherever you have higher productivity, like in upwelling zones or along the equator, you're going to have stronger food pulse signals than in this area, which we call the oligotrophic ocean, or nutrient poor. Yeah.
And so would those indicate like higher biodiversity in deeper depths up there, probably? Yeah,
I mean there's some hypotheses for how each of those is impacted. It has been suggested, and there's some data to support this, that in areas of very high productivity, like in upwelling zones, for example, off of the west coast of the US, or even off of the west coast of South America, very high productivity results in relatively low diversity, but high biomass, high abundance of species, where sometimes when you are in a more oligotrophic or nutrient poor environment, the scarcity of resources might result in more kind of niche partitioning and greater diversity, but perhaps lower abundance in certain places.
Interesting.
Because they're all competing for the same resources, right? They're all competing for marine snow. So, you know, if you can, you know, create a different way of competing and, you know, for that resource, you know, another resource is potentially space too, right? Space, you can see how a lot of these corals that are larger and more successful are perhaps the ones that are, you know, on the prime spot of the rock, right?
I haven't done that in a while.
Yeah.
Good spot.
Fascinating.
How'd I do that?
I don't really know. Maybe it was when I
But then
came
there are
around.
also other factors that might influence diversity patterns. For example, in the deep ocean, there are
tilt up with your camera.
Sorry.
Latitudinal gradients that somewhat mirror what we find in the shallow ocean. So we know that the equator and equatorial latitudes typically are the highest diversity of, you know, biodiversity for a lot of different animal groups. And as you move towards the poles, it's slightly lower. But in the deep sea, that trend may be similar, but there might be some pockets of difference.
I hate it when I do that.
Especially with corals, if we're just talking about coral diversity patterns, we would expect that if a lot of coral species, you know, were originally shallow water organisms and then invaded the deep sea, that they might actually have, you know, similar patterns that mirror that of their ancestral lineages. But there's a lot of other processes that could be going on in the deep ocean that might result in, you know, slight differences in those patterns. So it's hard to make generalizations about diversity in the deep sea without really doing these kinds of surveys where we're sampling and collecting and making, you know, conducting these analyses back in the lab.
Oh, wow. Fascinating.
So interesting. I love how, like, each time we continue to learn something new, it just inspires more questions.
Yeah.
I guess not, it just inspires more questions. Kind of the whole point is that it continues to inspire questions. The oceans are tremendously
It's right
complex.
below you. Land on top of us. Another 20.
It's right below you in your bubble cam now.
What's that?
It's in your bubble cam. Is that it there?
Yeah, no, we're there, but we need to move the ship another 20 meters east. Yes, please. Yeah, put it right on top of us. You think so? Okay. I don't quite have enough leash there. So.
No.
Yeah, let's move it. Even if it does come and goes on the other side behind us, that's all right.
So we've got about a 40-meter drift from the stern of Nautilus, eh?
So by, you can come down 5 meters.
Coming down 5.
Come down 10.
Coming down 10.
Live large.
Can you take a screen grab of this little landscape here?
Can.
Cool.
Oh, for those of viewers at home, if you were to switch to channel 2, I can see actually a really nice shot of the kind of coral area that Hercules is currently perched on.
Yeah, that is nice.
Ferdinand smelling the flowers again.
So also, while we get the ship all set up for the sampling, we do have another video highlight that dropped today on the Nautilus Live website. So if you want to check that out, click the gallery tab. So and check out the iridescent red ctenophore that we spotted on our second dive. Really beautiful look at the cilia. The cilia plates there, the coordinated rainbow flashing lights.
Is the starboard box accessible here? Because we're going to need to put the samples in there, I think.
Yeah. Should be it. Might be a little collateral damage there when we open it. I'm trying to get her to perch here.
Do you want the porch light on or off for this?
I'm not bothered right now. I'm just trying to get the vehicle to hold still. What's happening is
We're gonna get the next watch team totally set up.
I know.
Rob's giving me the he's giving me the eye here.
come on, Herc, find a marble.
All right, the feed may get a little quiet as we do watch handover for the next few minutes, but the 8 to 12 team will be here to take you through the next 4 hours and the 12 to 4s after that. And we will look forward to, we're planning to recover the dive at 4 a.m. local time, so in 8 hours from now. Lots more exploration left to come. And we'll be with you the next time we're back in the water. So keep those questions coming for the next watch team, and thanks so much for being here on Nautilus Live.
Okay, I'm going to hand this over to Josh and let him do it because I'm failing. I'm off. Thanks, everyone.
right. Josh, I'm going to stick around for this sampling event.
Okay, sounds good. Dan was kind of giving me a little bit of the rundown there, but maybe I'd like to hear it from you and make sure I'm clear.
Yep. So, you ready for a brief rundown?
Yeah.
Okay. So the plan is we are going to collect this sea star with the branch of coral that it's eating.
Okay.
They can be in the same, grab, or they can be separate. And that's going to go into one compartment,
Okay.
probably in the starboard box.
Okay.
And then we're going to collect maybe another smaller branch, like somewhere over here. So on the same colony, but something that hasn't been eaten.
Okay.
So that's going to be collection number 2, or 3, depending on if the star is 1 or 2 collections. And then for the final collection, we're going to collect a small branch from this uneaten colony off to the right-hand side, or to the left. It doesn't matter which one. They're both the same.
Okay. And then am I seeing that right with there's almost like a divide between that coral colony with the sea star, that the one on the left is eaten and the one on the right is not?
Yes.
It's
Yeah.
almost like down the middle sort of thing?
Yeah. So the eaten part is basically right here.
Yeah, okay.
We don't need a lot. We need maybe like 15 centimeters
Okay.
of linear tissue.
Sure.
And then the whole star, obviously.
The whole star, okay. Huh. So ideally, I'd like to be able to open the box and swing the arm around.
So Steve, this is one of your triad sample?
That's right.
It also seems very flat on the other side.
Yeah, the other side is not,
There's like no perching area.
There's nothing, yeah, nowhere to park. Try and be as gentle as I can.